Have you ever wondered what happens to the old worn-out parts inside your cells? Or how your cells digest the food they take in? It's like having a tiny, sophisticated recycling and waste disposal system running 24/7. Well, meet the unsung heroes responsible for this crucial job, the lysosomes. The name lysosome literally means splitting body. Lyso means splitting, and soma means body. And that's a perfect description for what they do. Picture them as small, spherical bubbles or vesicles enclosed by a single membrane. But don't let their simple appearance fool you. Inside these little spheres is a powerful cocktail of digestive enzymes, a veritable demolition crew for the cell. So, where do these powerful enzymes come from? The story begins in another part of the cell, the rough endoplasmic reticulum, or RER. This is where the enzymes are first synthesized. From there, they travel through the smooth endoplasmic reticulum to a cellular post office and packaging center known as the Golgi complex. Here, the enzymes are modified, sorted, and packaged. Finally, they are released from the other side of the Golgi, the transface, neatly wrapped in their own little membrane bubbles. These brand new vesicles, loaded with enzymes but not yet active, are called primary lysosomes. Interestingly, in plant and fungi cells, you won't find lysosomes in the same way you do in animal cells. Instead, some of their large vacuoles take on this role, performing a similar function of enzymatic hydrolysis. It's a great example of how different life forms solve similar problems in unique ways. Now, let's talk about the firepower inside a lysosome. They contain around 40 different types of potent hydrolytic enzymes. This isn't just one tool, it's a full toolbox. These enzymes can break down every major type of macromolecule in the cell: proteins, fats, carbohydrates, and nucleic acids. The environment inside the lysosome is also highly acidic, which is the optimal condition for these enzymes to work their magic. For a lysosome to do its job, it needs something to digest. This something is usually contained within another membrane-bound vesicle. These vesicles can be formed in a few different ways. Through a process called endocytosis, the cell can bring in substances from the outside. If it's a solid particle, like a bacterium, the process is called phagocytosis, and the vesicle is a phagosome. If the cell decides to recycle some of its own internal components, that's called autophagocytosis, creating an autophagosome. Once these vesicles, be it endosomes, phagosomes, or autophagosomes, are formed, they fuse with a primary lysosome. This fusion creates what we call a secondary lysosome. This is where the real action happens. Inside this newly formed digestive chamber, the powerful enzymes get to work, breaking down the complex molecules into their basic building blocks. Proteins are broken down into amino acids, carbohydrates into simple sugars like glucose, and so on. These valuable, smaller molecules are then transported out of the lysosome and back into the cytoplasm, where the cell can reuse them for building new structures or for energy. It's the ultimate cellular recycling program. So, what are the major functions of light? We can break it down into 4 key roles: intracellular digestion, autophagy, autolysis, and sometimes even releasing enzymes outside the cell. Let's start with intracellular digestion. Imagine a single-celled organism like an amoeba engulfing a food particle. This particle is enclosed in a vesicle called a food vacuole. The lysosome then fuses with this food vacuole, forming a secondary lysosome. The enzymes digest the food, and the useful nutrients are absorbed into the cell's cytoplasm. What's left over? The undigested waste is held in what's now called a contractile vacuole. This vacuole then moves to the cell membrane and fuses with it, releasing the waste outside the cell in a process called exocytosis. This entire sequence is intracellular digestion, a complete meal from start to finish, all within one cell. Next up is autophagy, which literally means self-eating. This might sound destructive, but it's an essential housekeeping process. Cells are constantly producing new organelles, and the old, damaged, or simply unneeded ones need to be cleared out. That's where autophagy comes in. The cell wraps the unwanted structure, say an old mitochondrion, in a membrane, creating an autophagosome. A lysosome then fuses with it and digests the contents, recycling the raw materials. This process is vital for maintaining cellular health. It can also be a survival mechanism. During starvation, a cell can use autophagy to break down less essential components to provide energy and building blocks for more critical functions. It's also a way for the cell to regulate the number of its organelles. For instance, if you start a heavy workout routine, your muscle cells will produce more mitochondria to meet the increased energy demand. But if you stop exercising, the cell doesn't need all those extra powerhouses. So, through autophagy, lysosomes will digest the excess mitochondria, bringing the number back to normal. It's an incredibly dynamic and responsive system. Now, for a more dramatic function, autolysis. Sometimes, for the good of the organism, a cell needs to be completely eliminated. This is a form of programmed cell death. During certain developmental processes, for example, tissues need to be remodeled, which means some cells have to go. In autolysis, the lysosomes within a cell will actually rupture, releasing their powerful digestive enzymes directly into the cytoplasm. This quickly breaks down the entire cell from the inside out. The resulting cellular fragments are then cleaned up by neighboring phagocytic cells. Because of this ability to trigger self-destruction, lysosomes are often nicknamed suicide bags. It's a drastic but necessary function for proper development and tissue maintenance. But what happens when this intricate system goes wrong? Since lysosomes rely on a whole suite of specific enzymes, if even one is missing or defective, things can get messy. When a specific lysosomal enzyme is absent, the substance it's supposed to break down can't be digested. Instead, it accumulates inside the lysosomes, causing them to swell and disrupt normal cell function. This leads to a group of conditions known as lysosomal storage diseases. These are genetic disorders, meaning they are hereditary and often present from birth. They can be incredibly serious, as the buildup of undigested material can damage cells and tissues throughout the body, particularly in the brain and nervous system. Scientists have identified over 20 different types of these diseases. A well-known example is Tay-Sachs disease. In individuals with Tay-Sachs, a crucial enzyme needed to break down a specific type of lipid or fat is missing. As a result, these lipids build up to toxic levels in the nerve cells of the brain. This accumulation causes severe and progressive damage, leading to devastating neurological symptoms. Sadly, most lysosomal storage diseases are fatal in early childhood, highlighting just how critical the proper function of these tiny organelles is for our health. So, the next time you think about your cells, remember the mighty lysosomes. They are the diligent recyclers, the meticulous housekeepers, and the disciplined demolition crew all rolled into one. From digesting a meal and recycling old parts to making the ultimate sacrifice for the greater good, these small vesicles play a massive role in keeping our cells and oozle live and healthy. They are a perfect example of the incredible complexity and efficiency that exists within even the smallest components of life. Thanks for watching. If you found this journey into the cell fascinating, don't forget to like, share, and subscribe for more deep dives into the world of biology. See you in the next video.